next speaker is Marcie Glicksman with Enclear Therapeutics. And the name of the presentation is called Device-Based Therapeutics for CNS disease. And I'll just do a little introduction on Marcie. For the last 30 years, she's been dedicated in developing better therapeutics for the nervous system and other therapeutic areas. Her efforts have resulted in 8 drugs entering the clinic, 2 marketed drugs resulting in over $2 billion in sales for Cephalon, now Teva Pharmaceuticals, and Cubis, now Merck. In addition to making impacts in the private sector, she has created multiple startup companies that raised over $40 million while at Harvard Medical School's Academic Drug Discovery Center. As a scientific leader, she has been a key advisor for multiple foundations, including the Michael J. Fox Foundation, the Alzheimer's Association, and the National Multiple Sclerosis Society. So with that, we'll introduce Marcie for her presentation. Thank you, Marcie.
Great, thank you. Anyway, thank you very much for the invitation, and I'm very excited to be able to present today the work that we're doing at Enclear. So if you go to the next slide. So just a summary of what I'm going to tell you. I'm going to tell you a little bit about our company and how we're focused on delivering therapeutics to the brain more effectively. Also, for those of you in the audience, most of you probably know the blood-brain barrier continues to be a challenge for CNS drug discovery. And so we use CSF flow, the flow of CSF, to have a more effective distribution of therapies into different parts of the brain. And then I'm going to show you some of our data, and we have additional data as well. But some of the data to show, these points. So the next slide. All right, great, thanks. So a little bit about Enclear. We were launched in 2018, so a fairly new company. We're developing our device as a health platform, CSF health platform, in order to improve and extend the lives of patients with CNS diseases. And so our business model is really to build an integrated neurospecialty pharma business, both with internal programs as well as partnerships, with a pipeline of applications for internally for neuro-oncology is what I'm going to talk about today. But we have other applications as well. And as I said, we're interested in additional partnerships. So if you feel free to reach out if you have an interest in speaking about that. And so if you go to the next slide. So this is just as background for people who may not be as familiar with the nervous system. But your brain is actually protected at 5 sites. So it's serious business to try to get things across the blood-brain barrier. There's the blood-brain barrier itself, which is probably the most well-known, but there's also blood-CSF barrier. There's an arachnoid barrier, and these are the cells that surround the outside of your brain. There's the blood-retinal barrier, so this is the barrier between your eyes and the brain. And then there's also blood-spinal cord barrier. So there's really a lot of protections, and that's for a good thing, because you want to minimize bad things, of course, getting into your brain. The next slide. So these all these protections, though, do allow some molecules to cross. So there's some transporter proteins. So some strategies have been to link proteins to some transporters that are known to get across the barriers. There's also specific transcytosis that happens. There's also paracellular pathway, but this is very difficult and pretty rare because molecules have to get through tight junctions. There's diffusion, and that's probably the most common. So if molecules have certain properties, it is possible for them to diffuse into the brain. And then there's an active P-glycoprotein pumps as well. So there are ways the brain has of getting things that are important back and forth into the brain. But the problem is that all other molecules are excluded. So when you're talking about therapeutics, this is a real problem. So the next slide. So also this is by way of background. These are some of the things that some people may be less familiar with. So this is just showing in this diagram in the middle. In the light blue is your CSF. So as you can see, it's around the outside of your brain. There are ventricles internal to your brain, and then it flows around your spinal cord as well. And so the arrows indicate the direction of CSF circulation. And CSF can also exit, and one of the areas it exits is through the arachnoid granulations, which are on the surface of the brain. So what Enclear system does is it administers therapeutics through the ventricle. It can also administer through the spinal cord. And I show on the far right side a cross-section of your brain, which also highlights the problem. So you see in the center, white area in the very center are your ventricles. So this is where CSF flows. And then it also, on the surface, you see the white area on the external part of your brain. So you can imagine that even if you give molecules in the ventricle, there's a lot of tissue that it has to penetrate through. And so a lot of areas, especially deeper brain structures, it's very hard to access when you just give therapies through conventional manners. So the next slide. So this is a list of some common methods for delivery that exist now. So some is just bioavailable molecules. So these would be small molecules that are whose properties are pretty well known in terms of likelihood to cross the blood-brain barrier. And so chemists are very familiar with this. So a molecule needs to be pretty lipophilic. It needs to be below a certain size to be able to get across. So if you give molecules intravenously, you may have as much as 1% get into the brain. So a small fraction will often get into the brain, but that also means that you have to give such large doses to be able to get enough to have an effect in the brain. So intranasal and submucosal are other areas that people have explored in terms of accessing, and there's certain properties that molecules have to have too in order for that to be successful. And again, only a portion will actually get into the brain from those regions. And then, as I said, actively transported proteins. So if you can link your molecule to something that's actively transported, for example, transferrin has been used. Sometimes this can also facilitate, and hopefully your molecule will still be active with this linking. And then also people do inject intrathecally and to the spinal cord, and people also inject directly intracerebral ventricularly. But as I said before, it can be problematic if you really need to get even distribution, or if you're trying to target certain regions of the brain. So all of these methods may get into the brain, but but are they really getting to the areas that are needed? Are they distributing evenly, if that's your goal? And what's the concentrations needed in all the areas? And then the other thing is, the dose-limiting peripheral side effects. So the fact that you can give things directly to the brain, you can limit the access your molecule has to the periphery that may generate some toxicity. But also we can control concentrations within the brain, and so that can also help limit even toxicities within the brain. The next slide. So here's a picture of our device. And so as you can see, it's got a catheter into the ventricle as well as a catheter into the spinal cord intrathecally. And the key thing is there's a pump. So we can actively move CSF fluid. And this actually facilitates the distribution quite a lot. And it means we have a number of ways we can control delivery. And this system can be used as a single dose, a one-time use. and just placed with local anesthetic. Or it can be used for multiple doses, and the system can be implantable. And then there are ports, access ports that are for people who need multiple doses of a therapy. They can go to just an infusion center to get additional doses once it's implanted. The next slide. So how, it seems like a lot to implant into a person, but in fact, the procedure that our system uses combines actually very common performed procedures. So, for example, putting a catheter into the ventricle, there are ventricular peritoneal shunts that are commonly done. There's about 30,000 performed every year. This is to relieve hydrocephalus. So it's a way of relieving the pressure from hydrocephalus. So it can be used acutely as well as chronically when this is an issue. It can be something that's implanted for a long period of time in adults, more than 8 years, without any issues. And the procedure is not new. It's been performed over 60 years. So we're combining that process with the intrathecal, putting a catheter intrathecally. And this, again, thousands of these are performed every year throughout the world. They're particularly used for management of pain and spasticity. For example, an epidural is given during childbirth, and I think people are very familiar with that. So this is also a common delivery method for oligonucleotides and AAV and brain cancer drugs to give them intrathecally, just as a standard of care as well. So this is so these procedures are done easily by surgeons. And the next slide. So what are our advantages? So as I mentioned, we can reduce peripheral toxicity. We can lower the drug dose that's needed, which can be important if it's expensive, but also helps reducing side effects. We can have less toxicity because we can actually control the concentration that tissue is being exposed to. We can also do bidirectional flow. So this can be a way of optimizing specific areas of the brain. Also, controlled release, like peristaltic type of delivery, can be used as well. Using this active CSF mechanism actually helps drive the diffusion into the tissue. So that's really one of the biggest benefits. In the next slide. So just a list of possible applications, peptides, RNA, DNA, large proteins, small molecules that penetrate the blood-brain barrier, but maybe you need to limit the distribution to avoid peripheral side effects. For example, antifungals and oncology drugs fall into this category. Any therapy that can benefit from repeated applications or slow infusions, our system could be used for this. Also, for example, for AAV, if you want to deliver it to the brain, you need to use AAV 7 or 9. But with this system, actually, you wouldn't be restricted. And then there's other applications as well. In the next slide. So I'm going to show you some data that we've generated. We've used sheep as a model. And so since a lot of people are not that familiar with sheep, they're actually pretty nice animals to work with. They're very docile. I've gotten a new appreciation for sheep. They live a good life on a farm somewhere, and so they're easy animals to work with. And I just wanted to sort of highlight differences when people are doing studies in different species. And when I'll give some examples also in some publications. When you compare mouse to sheep to monkey to human. You can see in terms of brain mass, sheep, monkey, and human. Sheep and monkey have about the same amount of brain mass, not quite as much as humans do, but much better than rodents. CSF volume, also sheep is actually getting closer to the volume of humans. And this is particularly important when you're talking about, delivering to CSF in terms of what concentration you're getting. And you can see monkey is actually fairly small. And then rodents, of course, are very small. CSF production, so when you're talking about being able to deliver molecules and keeping concentrations in CSF, your CSF is always being produced, and it's always turning over. And so this gives you a comparison among the different species in terms of that turnover rate. And so actually, number of turnovers per day is pretty similar between sheep, monkey, and humans. And then the brain features. So this is going to be important in terms of how good diffusion is across tissues. So sheep, monkey, and humans all have sulci and gyri, which are the infoldings on the outside of your brain, and of course mouse and rats do not. So we've done all of our initial studies in sheep, and actually we're going to just start doing some non-human primate work as well. So next slide. Sorry. I don't know, my phone has not rung all day, and now I'm getting phone calls. I apologize. This is, if you click on the bottom of this image, it should be a movie that will play. Yeah, thank you. So this is a experiment that we did with when we injected gadolinium into a sheep to look with our system, our circulation system, to see how it circulates. So as you can see, and the time frame is 4 hours. So as you can see from the beginning to the end, it's disappearing from the ventricles, which are in the center, but then reappearing. So it's very cool. It takes 4 hours for CSF to circulate with the pump at the speed that we did this experiment at. So you see the label appearing and then disappearing and then reappearing at the end of 4 hours as it circulates through the brain, through the spinal cord, and back up to the ventricles. And then you can also see how it's distributing into the tissue as well. So in the next slide. So this is so now I'm going to talk about our internal program that we have looking at cancer in the brain and how our system could be applied for helping, particularly this leptomeningeal carcinomatosis. So this is a cancer of the meninges, which is the lining, the pia mater and the subarachnoid space in the brain that lines the brain. And this is actually fairly common. It's about 110,000 new patients per year. And it's common in breast and lung cancer and melanoma. When those cancers metastasize, it often appears as this leptomeningeal carcinomatosis. And it's pretty sad, actually. These patients really have very little time to live once it's diagnosed, and very hard to control. So the current standard of care is to use a methotrexate or a similar drug delivered intravenously or intrathecally or intracerebroventrically, depending upon the physician. They can give it up to twice a week or once a month, again, depending upon the patient. And there's a lot of side effects, of course, with methotrexate in terms of peripheral toxicity. So there's really dose-limiting amounts that can be used. And it's also very inefficient at reaching all areas of the CNS, which I'll show you some data on. So we're hoping to be on the market in 30 months with this combination of methotrexate and our device, and to get good distribution therapy. And as I said, these patients have basically 2 to 7 months to live from the diagnosis. So we hope that with our system, we'll actually be able to extend the life of these patients and be able to give them a better quality of life longer. And the next slide. So on the right-hand side, I'm showing a graph. So this is a published study we didn't do, but just to highlight even the difference between giving methotrexate to these patients intrathecally in the spinal cord versus intracerebroventricularly. And the red is ICV into the ventricle, and the blue, the darker line, is giving it intrathecally. So you can see even that change alone actually improves survival for these patients. And this study was a retrospective study where they took 40 adult patients that had been diagnosed with LC. 30 patients had received chemotherapy through the lumbar puncture alone, and 10 of these patients had received it through intraventricularly. So even with the small patient size, you can actually see at least a shift. And so what we think is with our system, actually, we could get a better shift than that. The next slide. So this is data that we've generated in sheep. And so the far left side is when we've just given the methotrexate intrathecally in the lumbar region of the spinal cord, and then looked at different tissues of the brain and then also the spinal cord. So the two far right bars are the spinal cord. So the biggest bar there, the biggest blue bar, is the lumbar region. So that's where we're injecting it. So that's where you would expect the highest levels to be. And then the bar to the left of that is the cervical region of the spinal cord. And now it's even a lot lower, but actually at least you can still measure some. So it is going from the lumbar portion up to the spinal cord. But when you look at all the brain regions, it's not measurable at all. So it's very ineffective at reaching brain regions at all with methotrexate. And then the middle graph is the intracerebroventricular. So this is ICV alone, so also not with our system, and just standard of care ICV. And so you can see the far right is the spinal cord. So since you're injecting it into the ventricle, very little is reaching the spinal cord. And then you see the brain regions are really variable, but all on the whole, fairly low levels, and especially in deeper areas. And the far right is with our giving it into the ventricle, but with our system of circulation. And so you can see actually the levels are much better. And for example, even though we're injecting into the ventricle, you can see good concentrations in the spinal cord, which are the blue bars on the far right side. So the next slide. So I just wanted to touch on some other applications just in the literature, just to illustrate that not only do we have applications for cancer with methotrexate that we're developing, but it has other applications as well. And this really hit hard when there was the release recently from Roche and Wave Life Sciences, who'd run clinical trials on Huntington's disease. And they both have stopped their clinical efforts because of lack of efficacy. So again, at least part of what may have contributed to this was the not getting good distribution in the brain. And the next slide. And then this is also another publication and looking at different brain regions. And this is also applied to Huntington's disease. The top three graphs are looking at soluble mutant Huntington levels and looking for suppression of them. And then the lower three graphs are looking at aggregated mutant Huntington, and again, looking for suppression. So if you look in the striatum, for example, the first graphs on both rows, you're getting 15 to at maximum 30% suppression of the protein expression. So you're really having very small effects in this case. And then hippocampus, the higher dose. So L is low dose, H is higher dose. So you can see higher doses are getting a little more suppression in the hippocampus in terms of the aggregated mutant form. But really, in the cortex, you're just getting very modest suppression. In the next slide. This is another publication, actually interesting because they compared mouse with non-human primates. So in the upper graph is mouse, and the gray bars, the first two graphs are the controls, PBS and non-template control, and then the red is the data at two different levels in the mouse. So you get some suppression in some of the areas of the brain at the lower concentration, but not in the deeper brain structures like the putamen. In the deeper, I think they call it the medial cortex and the posterior cortex. But at higher doses, actually, you're getting good suppression in the mouse. But when you go on to the non-human primate, and you're looking at the caudate and the putamen, which really are the areas of most interest for Huntington's, on the far right, you're seeing that you're really not getting good decrease in protein expression in the non-human primate. So people are having to either give higher doses or give multiple injections to be able to attain anything that they think will be therapeutically meaningful. So these are applications that I think that our system would have a good chance at improving. And then the next slide. So this is just summarizing the points that I hope have come across, which are that there's a lot of barriers associated with crossing the blood-brain barrier and being able to deliver molecules effectively into the brain and getting good distribution throughout the brain. And that we think that our system has an advantage over this, and that we can give controlled and continuous and personalized delivery of molecules using our active CSF flow system, and we can optimize the dosage, and we can reduce the risk of peripheral side effects. And I think I'll stop there. Thank you for your attention.
Marcie, thank you. That was a great presentation. Just a quick question. So for the device, what is the regulatory pathway that you guys have to follow?
So we're working that out in some discussions with the FDA, but probably de novo. It'd be a de novo use.
Great. No, that seems like a fantastic device, and the data is there to demonstrate the potential.
Yeah, thank you. Yeah, we're very excited about it. We think it's especially these times of where gene therapies are so exciting. To be able to have a better way of delivering, I think is going to be important for patients.
Yeah, for sure. Sam, I don't know. I don't see the questions. Do you see any?
Not at this moment. Again, I do want to thank Dr. Marcie for presenting. It's always a pleasure to hear from you.
Thank you. Yeah, and if anybody in the audience wants to reach out to me, that would be great. We can have further discussions, and if there are more questions, happy to talk to people. Thank you.
You're welcome. And we'll head over now to our next talk by the tag team presentation from Rapid Novar. So let's all head over to the next talk.